Hello, we will start explaining Synapse over here. So the first thing we have to know in Synapse is the structure, the names of the structure. So let's start looking at this diagram over here to get the names. We might be asked to label the diagrams having this synapse and all that. So the first thing we have to know is synaptic knob, which is this whole region over here, this one, okay, this whole region. These are the synaptic knobs. We have two synaptic knobs. There is this presynaptic knob and there is postsynaptic knob. Presynaptic knob means the region from where it is coming from, and then the region where it's getting in is the post. Okay, so that's the first thing. And then let's look at the other diagram, which is the synaptic vesicle, which is this part over here. Then we have this region between the two between these two synaptic knobs, which is called synaptic cleft. Okay, so that must be noted. And then we have these neurotransmitters, which are just chemicals inside this synaptic vesicle. And lastly, we might have some other, but they are not labeled here. We should know that it starts from the axon over here. This is the axon region, this one, as you can see over here. This is the axon region. And then we have this the dendrites of the adjacent neurons, and this is the terminal of the neuron. You should know that this is the beginning of another neuron. So that's the first thing we're supposed to know. And then after this, we are supposed to know how the impulse move over here. So we know that when an impulse starts from here somewhere, then it moves here, but then over here it has to pass through the synaptic cleft to the other side. So how does it pass over here? So it's a simple process actually. We just have to bear in mind some few ideas. So the action potential gets here. Action potential simply means that the sodium ions enter here, and the voltage is increasing, and so these calcium ions from outside enters inside. Okay, so the first thing is this calcium enters. Okay, the calcium enters, and this calcium, so we can list them. The first thing is calcium enters. Okay, this is 2 plus. Calcium enters. Then we can go to the second thing, which is calcium 2 plus max synaptic vesicle fuse. Okay, that's simply the second thing. So the first calcium enters, then synaptic vesicle fuses with this region over here. Okay, so it fuses. This fusing is caused by calcium. Okay, then the vesicle pours its material outside. This material is called the neurotransmitter. Now in normal circumstances, like most of the neurotransmitters in our cells, in our neurons, at the synapse are acetylcholine. These are normal ones. Then there are others as well. For example, there is a list over here. We have noradrenaline, we have this one, dopamine and glutamic acid, which is in the brain. So you might be asked that to explain about cholinergic synapse, and if you are asked about cholinergic synapse, then you should strictly adhere to acetylcholine. You shouldn't write just neurotransmitter, rather you should write acetylcholine. Okay, so let's consider the acetylcholine case, because this is the thing labeled here. So this neurotransmitter are poured to the adjacent neuron. Okay, this is the other through the synaptic cleft. We know that this gap over here, this one is synaptic cleft. This whole region between here, this is a synaptic cleft. Okay, so then the neurotransmitter enters here. Once it's inside here in the other neuron, like we can say it's this region here in the other diagram. Okay, when it is in this region, an interesting thing happens over here. We should be very careful about this. These ones enters and sticks to the protein channels. You see over here. So when it sticks to the protein channel, this allows sodium to be released. So sodiums get released, sodium go out, and that's how an action potential. No, actually, it doesn't go out. Sodium, sodium are moved inside, and they are not moved this way. Actually, they are moved this way, and this sodium, when they are already over here, this causes depolarization, and we have an impulse created over here, an action potential. Okay, so again, to summarize this part, it might be a bit confusing. Calcium ions, action potential reaches this region here. Calcium ions, calcium ions are diffused in. Calcium ion causes the synaptic vesicles to stick to the membrane here. The synaptic vesicle releases its contents, which are the acetylcholine, the neurotransmitters. The neurotransmitter enter the adjacent neuron, and over here they allow sodium ions to flood through the open channels over here in the postsynaptic membrane. And when the sodium ions flood inside here, this simply creates an action potential, depolarization, which causes sodium to flood in and potassium to flood out, like this here. So this automatically creates a potential, after which this sodium will be moving this way. So it will be the same sodium which was coming here, now it will be moving here. So this is a similarity, like it's the same process going on. And you can see in other diagram as well. This is the axon, which is this part. It comes here. These are the synaptic vesicle. They are attached to the presynaptic membrane by the action of calcium ions, and they pour their neurotransmitters. The neurotransmitter are attached to the protein channels, which allow the sodium ions to sort of accumulate over here, and this then causes sodium ions to move as an impulse. So sodium starts here, so we have a sodium here. Okay, we have sodium here, and then we get sodium here as well. And what causes this? There is this calcium getting in, which causes this whole process. Okay, so we should be very careful with this part. So this is the first video, and I'll make the second video to emphasize. Maybe we should just finish the last part of this, which is what happens to the neurotransmitters then. So you should know that the neurotransmitter goes in this side, but then there is this acetylcholinesterase enzyme in this region over here, okay, which sort of changes acetylcholine into acetate and choline. Okay, so the acetylcholine which comes out from here, it is poured inside here. It is sort of changed to acetate and choline by the acetylcholinesterase enzyme, and then these acetate and choline are produced over here, and choline moves back to the neurotransmitter. I mean, moves back sort of diffuses back like this. So acetylcholine sort of moves back like this, and sorry, choline moves back like this. So we should name it here so that we can not get confused. Choline moves back here, and then this reaction comes into play. Choline, there is coenzyme over here, there is coenzyme in this region which reacts with choline and gives you acetylcholine back. So acetylcholine again forms a vesicle like this, and then again is released. So it is like a continuous process. This is like recycling of the acetylcholine. When it goes back here, and then it breaks down, and then it moves back, choline moves back, and then it attaches itself to acetyl. It attaches itself to coenzyme A, and it again moves back here. So this is like a continuous cycle kind of thing. I think we should better finish this over here only. Then so what are the roles of this synapse? Okay, why do we need a synapse in between two neurons? Why is there not a continuous continuous transfer of impulse? Like, okay, so one of the reason we have synapses because it ensures one-way transmission. It's like this synaptic vesicle can break down and move and pour material only on one side. It can't go the opposite side. Like, we can't have an impulse starting from here and moving this way. It's impossible. So we sort of get this one direction, which ensures that the intended direction, like the impulse where it's required, it reaches at the right time. It doesn't meander somewhere else. And then we have this possible range of action in response to a stimulus. This is a very good reason because there is this random connection in the impulse kind of thing, and if there is this different impulses going to different regions, and if there could be this neurotransmitters, there could be this there couldn't be sensory, there couldn't be synapse. The connection between neurons would be very sort of complicated, like, you know, it would be very limited. So this synapse, we have to know that one neuron can have many synapses, so it can get information from many other neurons. Okay, so that's the reason we have this synapse, and that's the reason they are very important. And then it is involved in learning and memory. And the last thing we should understand is it has a role in when we talk about nicotine in the cigarettes and all that, we sort of deal with the synapse over here. What happens when we take in a cigarette, when we take in nicotine? Nicotine is a neurotransmitter, and it sort of makes this process of action potential to be longer, so we get this longer impulse over here. So it sort of gets more stimulation than the normal stimulation, and that gives the pleasure to the muscles and all that. And that's why it's very addictive kind of thing. And then we have this organophosphorus insecticide. This one is used as an insecticide by farmers and all that, and is very harmful. It inhibits action of acetylcholine steroids. So normally, if it is used in small quantity, it has no problem. But then continuous use of such chemicals can cause a problem in the nervous system. So that's all about synapse. Thank you for listening, and hope you understood it well. And if there is any question, you may post, and you can subscribe to my channel. Thank you.